Today we are going to talk about structure of chromosome. So in non-dividing state, that means resting state, chromosome stays as single strand. When cell is ready to divide, each chromosome strand duplicate and form two sister chromatin. Okay. Now you see there is a short arm and a long arm. Short arm we name as P and long arm as Q. You can remember in the alphabetical order P is coming before Q, PQ. So short one is P, long arm is Q. Then see the centromere, that is in the middle part, you see in red color, that is a centromere. When this chromosome make two sister chromatid, those sister chromatid attach to each other at the centromere. Okay, it's only seen when it is about to divide, okay, when they prepare for the cell division. Okay. Now the terminal end of the chromosome, we call it telomere, and some chromosomes other than the telomere have an additional part called satellite. Okay, satellite is not the other satellite we are here talking about. It's about the satellite in the chromosome. It's a highly repetitive non-coding DNA segment. Usually adenine and thymine, 80 sequences are repeatedly there in thousands or millions times. Okay. Then the satellite is attached to the chromosome N with a stalk. It's a satellite stalk. We call it secondary constrictor. Why we call this secondary constrictor? Because the centromere, we can mention it as a primary constrictor. Okay. Now, do all chromosomes have satellite? No, it only present in some chromosome. Now let's see a chromosome with sister chromatid attached at the centromere. And what is the function of the centromere? It connects two sister chromatid. Other function is it binds to the microtubule of the mitotic spindle via the kinetochore. Okay, so centromere is underneath the kinetochore. Kinetochore is that binding protein. Kinetochore is a disc-shaped protein structure. It links with the microtubule of mitotic spindle in meiosis and the mitosis. Okay, if we talk about the centromere, that is underneath the kinetochore, it consists of alpha satellite DNA. The name is alpha satellite. The other satellite we had previously don't contain the alpha satellite, but here centromere have alpha satellite DNA. Those are randomly repeating non-coding DNA with 170 base pair repeat unit. It mainly consists of heterochromatin. You remember heterochromatin, right? It's a gene poor area. Heterochromatin is a gene poor area. Okay, now I would like to tell more about telomere. It contains thousands of TTA GGG repeat. Okay, it's a protective mechanism for the chromosome DNA because it prevents from damaging. Okay, there are some protective mechanisms there. It ensures integrity of the chromosome by preventing the protective mechanisms is it prevents the DNA repair system mistaking the end as a double strand break. They do by forming the T-loop and the sheltering complex. So otherwise, if it is not there, DNA repair system will get it as a mistake and will think it's a double strand break and they try to repair, and that lead to progressive damage. So to prevent that, what they do is they make a T-loop and the sheltering complex there. Now I am going to talk about secondary constriction or satellite stalk. I think you remember NOR region, nucleolus organizer region, that we talked about in a previous video that we saw with the chromosome 13, 14, 15, 21 and 22. That is mainly acrocentric chromosome. The acrocentric chromosome term I'll explaining later. So this NOR region have repetitive heterochromatic DNA but with rRNA gene. You know heterochromatic DNA is have gene poor area, but particularly this part has rRNA gene. Comparatively, we talk about other regions. Those other regions have non-coding repetitive regions, right? But here this region has rRNA related genes. So next video we'll talk about metacentric, submetacentric and acrocentric chromosome.